So there was a question on the test that asked you, if you saw this, if this whatever is this color, which of the colors does this thing absorb? It absorbs all the colors except for that color which you see, right? I've repeated that. There were two colors on the test. But in this case, what you're looking at is one color, and those purple bacteria that they talked about in the video that gained energy. It's kind of some of the earliest forms of life. There was probably an even simpler form of life. Bacteria, I don't know if there's any evidence for it, but it probably did not depend on light. It was probably doing what's called chemosynthesis. No, you didn't. You didn't see chemosynthesis. You saw something else that looked like that. Yeah. But I did tell you about chemosynthesis in the hopes of trying to give you a clue to get a right answer on the exam, and you kind of missed the boat on that one. A lot of people did anyways. So with chemosynthesis, the idea is that you are not using the sun, you're using chemistry to make energy. And there's still some organisms on Earth today that use this chemosynthesis to produce energy. So here's chemosynthesis, and over here these started doing photosynthesis, and they were much better. Photosynthesis was much better at producing energy. So these little bacteria, remember they're single-celled prokaryotes, they don't have a nucleus, et cetera, et cetera. Now, interestingly, one of the questions that the test asked you, and I was surprised that so many of you got wrong, was that you have this bacteria, photosynthesizing bacteria, purple bacteria, right, that the video talked about, and it had photosystem 2, excuse me. But not number 1, not photosystem 1. But then this other bacteria, there was a green bacteria, had both photosystem 1 2 and photosystem 1. So then the question came up, which of the two were ancestral, right? That was the question on the test, and I told you ancestral means something that you inherited. So which of these two photosystems would you have inherited? You said 1, why?
Because I thought that since it didn't show up anymore, that means that the other one.
Well, here's the thing. For you to inherit something, so there's inherited, right? The stuff that you got from your ancestors. That means your ancestors had to have had it. That's right. They had to have had it. So in order for you to have inherited one of these two, the ancestor had to have had it. Right? So, photosystem 2 was the one that was inherited because this one had photosystem 2, but it didn't have photosystem 1. So you can't inherit something unless your ancestors had it, right? So where did photosystem 1 come from then if they didn't inherit it? Evolution. So it evolved.
See, that was confusing. You were saying why did photo 1...
I don't want to spend too much time on these questions because I already made a video, and during advisory I'm going to release it to you. The only reason I haven't released the video already is because there's a couple kids that haven't taken a test yet, or they haven't finished taking a test. They had to leave early. So what we have here is photosystem 2 and photosystem 1. The question was, let me pause this so I can find the question. Okay, so I found the question. It says some photosynthetic bacteria, purple sulfur bacteria, right, have photosystem 1 but not 2. Oh, so they did have 1, not 2. I reversed it in this example, but regardless. While others, cyanobacteria, have photosystem 1 and 2. All right. So which of the following might this observation imply? So let me correct this and say that in the problem, right, they said that the green, well, actually I had that part right. That the purple one had photosystem 1, but not 2, and the green one had 2 and 1. All right, 2 and 1. All right. So that explains maybe that explains why we have number 2 first, because actually number 1 evolved first, right? And then 2 came later. Even though it comes before 1 in the process, it evolved second. Does that make sense? All right, so the question is, which of the following might this observation imply? A said photosystem 2 might have been selected against by some species. If photosystem 2, in order to select against, what does that mean, select against? Anybody remember from middle school or in previous observations, anything about evolution? To select against, what does that mean if I select against? Natural selection is correct, yeah. But to select against, just tell me what you think it means. Listen to the words, select against. Right, what does that mean? You're not going to choose it, right? You're not going to choose it. So when you talk about natural selection, by the way, let me just put this one word out there and always remember it anytime you hear about evolution or natural selection. Here's the word that has to come to mind: death. Death. Something dies. So when you select against, what does that mean? You're killing it. You're killing it. It's gone. All right? All right. If you're a white rabbit, if you're a black rabbit living in the snow and there's a bunch of wolves around, what's going to be selected against? You're dead, right? Why? Because the wolves are going to pick you. Why are they going to pick you? Because they see you, right? So the things is selection, natural selection is all about who gets chosen to, right? And of course, if you die, do you get to have babies? No, if there's no babies, your genes go to the next generation? No. So nobody can inherit your stuff, right? Because you're dead. Does that make sense? Yeah. So when we're talking about photosystem 2 and photosystem 1, the question says photosystem 2 must have been selected against in some species. But did a species have, did any of the ancestors have to? So how can it be selected against? How can some predator choose to eat it? Or some natural event choose to kill it because of photosystem 2 if they never had photosystem 2. So that's why letter A didn't make sense. By the way, this is probably the only question I would say if you got wrong, and I say that in the video that I sent out, the only question if you got wrong, you had no, you had I was like, it's okay if they got it wrong because they didn't really get a lot of work on this question. The other questions, shame. I'm kidding. Yeah.
I thought about selected again. I thought it meant only certain ones got it.
Yeah, that's what I thought too.
Yeah, that's what it means. Only certain, well.
But I didn't know it meant like they died.
I didn't think that meant death.
Oh, okay. Well, then that's okay. You got that one wrong. I curved the test, right? That's why I curved tests. Because maybe you missed something because you didn't really get a lot of work on it, right? All right, so the next one, so just know that that's not possible. You can't, so know that selection, natural selection means you're killing it, right? So that whatever, it's being chosen for death. Whether it be because it's not fast enough and an earthquake kills it, or it's because it only eats eucalyptus leaves and all the eucalyptus leaves die so that they starve to death, right? Or if it's that they're a color that makes them easily identifiable by a predator, so they die more often, right? Or whatever it is. It could be sexual selection. It could be that the female chooses the most colorful ones, and these poor souls don't have a lot of color. For instance, peacocks. Why are their feathers so beautifully colored? Because they attract females. So if the ones that aren't beautifully colored, guess what? Oh, babies. They don't die. They get to live their lives, no problem. But they don't have any babies. No baby, no play, no babies into the future. Their genes don't go on. Please don't. Huh?
So it's kind of like a health disparity.
Well, let's not, that's kind of it's a little bit off topic. We'll just go ahead and yes, in a way, kind of. Maybe? All right, let's move on. So photosystem 2 may have evolved to be more photoprotective What does that even mean, photoprotective? Anybody know what that means? Is that a word we talked about? Is that even a possibility? Is that something that maybe you remember we talked about but we didn't mention it? No, that's something, that was what we call in test taking a distractor. This test was full of distractors. That's why you freaked out. That's why most people didn't do well, because there were distractors. And if you weren't confident in your answer, if you weren't sure about what was going on, guess what happened? You got it wrong. Right? So distractors are out there. They're predicting that this is a standardized exam. These questions are given, not made by me. I'm not the evil genius that came up with these questions. These are made by the state of Ohio, by the ACT people. These are the questions that you have to face so that you can be successful and get college money and be great. Yay, and be the next president of the United States or whatever it is. All right. So photosystem 2 may have evolved to be more photoprotective. That's just something they threw in there. It doesn't make sense. Cyclic flow must be more preventative than linear flow of electrons. It sounds fancy, doesn't it? It sounds fancy. Ooh, cyclic flow must be more. What? That's wrong. That was a confusing. It's a distractor. It's a distractor. If you stay to your fundamentals, please don't talk to each other while I'm trying to instruct you. I like how they look around. The two that are talking are the ones that are looking around like, unbelievable. So anyway, cyclic flow must be the most necessary of the two processes. What? When did we talk about cyclic flow? So, but you know how many of you chose cyclic flow? You chose it because it sounded right. Cycle. There's a bunch of cycles in there, but one of them. What does that mean? You didn't really know what you were doing. Because if you knew what you were doing, you wouldn't have chose D or E. You wouldn't have chose C probably, and maybe you chose A or B because you were confused and we didn't really talk too much about, B, photosystem 1 must be more ancestral. Photosystem 1 was older. And how do we know it was older? Because it was there, it's in both. Whatever's in both probably was there first. Right? Go away. Go away. I'm lecturing. You're interrupting. I don't understand. All right. So I didn't want to spend the whole day on that. I just wanted to touch on that one because that one was I felt the worst about. This is the only question I felt bad on. I shouldn't have put it in there maybe, but I thought it was challenging and you could have answered it and a lot of people did get it right, so it's possible. But you're right. We didn't really talk a lot about natural selection. We really didn't define it, so it wasn't a fair question. But it's okay. It was curved, like I said, so. I think the bigger problem, and the biggest problem on the test, is that you guys don't understand photosynthesis. I think that's the biggest problem, I'm sorry to say. And there's a second big problem, and they're both equally big, I think. You get distracted really easily by questions that sound fancy. If the question sounds fancy, you want to choose it. Don't do that. I know one student came up to me and literally told me the right answer, figured it out. She sat there and said, but this is true and this is true, and therefore this has to be the answer, right? And she's standing there looking at me, and I wanted to say, yes, you're right, go sit down, be good. But I can't do that, right? So I had to sit there and say, think about it, have confidence in yourself. So, she came up here and she was just like, wow. And she literally explained every right point. She understood photosynthesis verbally. She was explaining. I don't know if she chose the right. Did you choose the right answer? Yeah. Okay, so you did choose the right answer. But you almost chose the wrong answer, right? Yeah. And that happens so often in this class. Honestly, that's the biggest problem. Two problems. One, you didn't prepare enough. Remember what I told you about test taking. You have to remember you have stress. You have to remember that there's distractors on exams, that things that sound right even though they're not. You have to remember you have to read the questions. You have to remember you have to knock out the wrong answers. Don't just pick one. But also you have to prepare to the point where it's like, even though all that's happening, I'm still going to get an A. or a B on a test because I'm gonna overcome those things because I prepared so well, right? That's the trick to these exams. All right. So that was photosynthesis, and remember, chemosynthesis, that was one of the answers, right? I think that was the autotroph question that you came up and asked me about, right? I think it was, so you had autotrophs are both chemotrophs, chemosynthesis and photosynthesis are both autotrophs. They both make their own food. Heterotrophs eat the ones, so the only ones that are absolutely necessary for any ecosystem, for any living system, is an autotroph, right? And some nutrients. After that, if there's heterotrophs, great. If not, now long term, it's probably better you have heterotrophs because there'd be some feedback going on, but realistically, all you really need is an autotroph and you're good. Water, carbon dioxide, and photosynthesizer, or some chemicals, whatever the chemosynthesizer needs, and some water, and you're ready to go. All right. So that was the answer to that question. All right, so first life on Earth, and by the way, these guys here, these purple bacteria, one kind, these green ones, oops. What is this? Okay. These green ones are cyanobacteria. There were questions on the practice exam, and some kids told me there were some questions. I don't know what the questions are because I don't want to know what questions they give you unless the state releases them. I don't want to have any knowledge about that test. But some kids, it's illegal. I don't want anyone saying I cheated, right? Does that make sense? So like I tell you when you're taking a test, separate yourselves. Make sure you're not looking at anyone else's paper because I don't want anybody saying you're cheating, right? I never want anybody saying that I'm cheating. So I don't want to know what the questions are. But some kids were telling me that they asked about the purple and green bacteria on the test last year, an end of course exam. So keep an eye out for that. Everything I teach you is because I think it could be on that test. Does that make sense? If I don't teach it, there's a lot of stuff I'm not teaching you. And if I don't teach it, it's because we don't really have a lot of time, or I don't think it's going to be on the test, so I really want you to focus on what I want you to focus on. Some people don't have notebooks out, some people came late. I understand why you're coming late, but you should have binders out, you should be taking notes. Endosymbiosis was another one that people got confused on the test. We had covered it. Yeah, we talked about it, and we talked about it in August. I talked about it again during organelles. You read about it through your cell project, right? And it's this idea that there's this eukaryotic cell, right, or this big cell. Let's just call it big cell. Could have a eukaryote. Probably was a eukaryote. Who knows? It was sitting there, big cell, and it came around and found. Now this bacteria here was really good. And let me shrink all this here. So let's talk just about this. So this is back. There's this cell living back in the oceans, back in the day, before any people, before plants, before animals. Just this cell, right? Did it have a nucleus? Maybe. I don't know what the. I don't know enough to tell you what if it was a eukaryote or not. But let's call it a eukaryote if you don't mind. All right, you can look it up later. So it's a eukaryote, right? It has some stuff, but they didn't have mitochondria. They weren't really good at processing energy. This little bacteria here was really good at making ATP. It can make ATP really well. But you know what? This cell could move around really well. This one couldn't move around very well. That's right. So over time, what happened was this big one came along. Of course, it was really good at eating. It could eat really well. But it couldn't make ATP very well. Well, this cell, phagocytose, remember that word, right? You should know that word, which means it surrounded the cell, right? Look at the arrows. These are steps, right? And surrounded the arrow. And guess what? It brought that, and again, there's a nucleus. And it brought it in, and here's this. And of course, I'm going to go ahead and make this membrane blue, if you don't mind, just to show you the double membrane. And so that prokaryote was now inside that cell. So this thing could make ATP really well, and this one was really good at finding food. It moved around well. It did something really well, right? When it ate this, it was trying to eat this bacteria. It wasn't trying to like, hey, you want to make friends? No, it was trying to eat it. But something about that bacteria didn't allow this big cell to digest it. So it just sat there. Of course, it was still alive. Well, some of the food, some of the nutrients went diffused into that cell. And that cell made a lot of ATP. So much ATP that some of it came back out. So what did this cell get? And ATP, right? This cell got ATP. And what did the bacterial cell get that was in the double membrane? Why is there two membranes?
Because when it enters it, the plasma membrane coats it.
That's right. So when it enters it, this plasma membrane makes a vesicle. This is part of that vesicle. So this bacteria got sugar from whatever this thing ate, right? And proteins or whatever this thing ate, right? And was able to make a lot of ATP. So much ATP, it never had this much sugar because it wasn't good at getting it, right? So now it made so much ATP that this cell got an advantage. This cell was getting an advantage, right? Then you got that, when you have this kind of close relationship, what do we call it? A close relationship is not necessarily a good one or a bad one. A lot of people think symbiosis means good relationship. That's not necessarily true. Symbiosis could mean a bad one. It just means close, all right? So write that down. Because people were asking me, it really upset me when people asked me during the test what synthesis meant. I was ready to explode when someone asked me that question. I didn't. I give myself, pat myself on the back that I did not explode because I was like, are you kidding me?
Can you repeat that again?
Symbiosis means close relationship, right? Close relationship. So this word here, symbiosis, means close relationship. Could be good or bad. Right? So endo means what? What does endo mean? In, right? So this is inside. So why do we call this an endosymbiotic relationship? Endosymbiosis. They're so close that they're inside. All right, so this was really good at making ATP. It was it used oxygen to do it. It broke down sugar, or at least pyruvate at some point. Who knows how the steps evolved later on? Because we're talking millions of years. We don't really understand. There's a lot of evidence in why we think this is true. Because here's the evidence. Are you ready? Mitochondria, our mitochondria today. Mitochondria have bacteria like DNA. Their own separate DNA that's round, like bacteria. Their ribosomes, they have their own ribosomes. Bacteria like ribosomes. They're small ribosomes versus the big eukaryotic ribosomes. So isn't that interesting? That's the evidence that's part of the evidence. There's many more. Some of the genes are very close to the bacterial cells. So there's a lot of other evidence, but these are the two big pieces of it. Also, one more, there's a double membrane. Why do we think because there's a double membrane that would mean that, how would that support this idea of endosymbiosis? Because it got eaten, and then the plasma membrane of the hunter surrounded it because of phagocytosis, and you had two membranes, and that allowed this bacterial cell, to develop folds, right? Which increased surface area, which also increased the ability to separate high from low, and then you got a lot of ATP, and so this thing became better and better at making ATP. And so, and now today, we are one creature. We are one. You have every cell of your body has these little bacterial, these ancient ancestral, right, organelles that were at one point, we think, separate living creatures, right? But now they're just part of us. Every creature on the planet, every eukaryotic cell on the planet has these mitochondria. Isn't that interesting? So this must have happened really, really early, right? So now, interestingly, what I had told you before and why I thought that question was fair, that photosynthesis 2 and 1 was fair, even though it was really hard, and I don't mind that you got it wrong, if it made you think, is that you had in plant cells, what do you have in plant cells? You have the cell wall, you have the cell membrane, you got a nucleus, right? You got the endoplasmic reticulum, rough and smooth, and you have the Golgi body, right? But you have mitochondria, you do have mitochondria, and what do you have? What else do you have in there? Chloroplasts. So this one has chloroplasts and it has mitochondria. By the way, chloroplasts also have mitochondrial-like DNA, bacterial-like DNA. They also have bacterial-like ribosomes. They also have double membranes, right? The thylakoid membranes and the outer membrane, right? So they have all these kind of characteristics as well. So what do we think about chloroplasts? They weren't mitochondria, but they were these bacterial cells that could do photosynthesis really well, where the bacteria or the mitochondria were these bacteria that could do respiration really well. So one could do respiration really well, the other one could do photosynthesis very well. So which one came first? Animals, or the mitochondria eukaryotes, or the chloroplasts eukaryotes, the photosynthesizing eukaryotes? The mitochondrial eukaryotes, right? The ones that let's not call them animals, because I'm not sure you can call them animals, but the heterotrophic were probably first, right? Because they had mitochondria, because you have, these plants have both. If these have both, probably, and by the way, their DNA is very similar to our DNA in the mitochondria. Our mitochondrial DNAs to plant mitochondrial DNAs are very similar, and you can do RNA studies and DNA studies and show incredible similarity between them. So we know they're probably related, okay? And so, since both of them have mitochondria, and only plants have chloroplasts containing eukaryotic cells were probably second, right? So one of these came along and took up the chloroplasts and became kind of the ancestor of, one of these picked up what we now call chloroplasts and became the ancestor of plants. Yeah, that's right. So that was endosymbiosis. So animals and plants. Now let's just talk just photosynthesis. Now we talked about photosynthesis. You know you got a second shot at this test on Friday. But let's go ahead and talk about what you did your homework, hopefully. I'm going to collect it today on your way out the door. Make sure you don't even pull it out now. I'll give you 5 minutes at the end of the period to make sure you get it all together, staple it, whatever you need. I'm not dealing with it. Chapter 8, let's not get into it, please. Oh, wow. So, both animals and plants do cellular respiration. They both do it in the same way.
No.
Yeah, they kind of do cellular respiration the same way. The plants do photosynthesis, but they do cellular respiration like you do. They have mitochondria. They do the same processes, right? So there are some basic steps you should know in order to be able to. What's that? Oh, jeez. There's some basic steps you should know in order to do well on a test on Friday, and it really shouldn't be that difficult. But then again, I didn't think you would do as badly on this last test. So it's all about preparation. I'm here after school today if you need some tutoring. All right, but let's go on. If you did the homework and you really did it and you understood it, then the questions should be clear, unless your distractors are bothering you. So there is a cell. Both animals and plants have cell membranes. Obviously plants have cell walls, right? But we're not going to worry about cell walls right now because we're just going to worry about just the cell membrane and what's inside it. They both have nuclei. The chloroplasts and mitochondria is what plants have, right? Where Animals have mitochondria. So we're not going to worry about the chloroplasts right now. We're going to worry about the mitochondria. The mitochondria has these folds, these internal membranes that look something like that. And that's an internal membrane. And whenever you have a membrane, what do you think is going to be happening? What are we going to be doing? What are we going to be doing? If we have a membrane, we're going to be doing what? What do membranes do?
The stuff come through and stuff.
They come through, transport, that's right. But what is number one thing membranes do? What's the number one purpose? Separate things, that's right. We're going to be separating, preferably from high and low. We'll probably have some high on one side, some low on the other. Something, right? And yes, because of that, you're going to be transporting across that membrane, right? So that's why we've been kind of pushing that. So you have this mitochondria. And this thing, it needs sugar, right? Sugar is the source of energy. We know that because plants, we just spent like a week and a half, 2 weeks going over how you get the sun's energy and make it into sugar. That's where the energy is. It's in sugar. It's in C6H12O6. It looks like this, a hexagon. We've drawn it. We've looked at the structure. We've looked at the atomic, the atoms, the elements involved, their position. We know that there's an oxygen at the 0 point, right? We know there's one carbon, 1, 2, 3, 4, 5, 6, right? So we know all that's happening. But we have to take this and we have to make ATP. How is that going to happen? We need ATP. We don't need sugar. Sugar doesn't help us. This is great. It's great that there's a lot of energy in this thing, but it doesn't help us because we got to get energy in what form? ATP. I'm going to make this analogy again. I made it before. If you have 1 million potatoes, those are valuable, right? But you can't buy a house with 1 million potatoes. What do you got to do with those 1 million potatoes? Sell them. You got to somehow convert them into cash. Right? I don't care if you cut them up and cook them into French fries and sell the French fries. If you sell the potatoes as a bunch. If you bake them and you make mashed potatoes and you sell the mashed. I don't care what you do. You got to take those potatoes and turn them somehow into green cash for you to make some money, so you can buy the house. Right? So this is the same thing that's happening here. You got this sugar molecule. It's sitting there. It's great. It's valuable. It has a lot of energy. But what do you got to do? Turn it into ATP. So that's really as simple as I can put it. Now, what's the first? It's sitting here outside the cell, the sugar. It got digested, or maybe it's floating around, or it's breaking down. It's broken down. So what do we do? What's the first step to taking this sugar and making ATP? Well, no, the sun, it's been made already. The sun's rays would be important, yes. If we were trying to, the sun's rays would be important if we were trying to make the sugar. But the sugar's made, it's sitting there. What are we going to do with it? Break it up. But what is the first thing? There it is, outside the cell. You're the cell. What do you got to do first? Come on, think. Bring it in. You got to bring it in.
It's gonna trap it, right?
It's going to trap it. That's exactly right. Excellent. Yes. That's it. Well, it's not. It's actually a carrier protein. It's not phagocytosis. This is a molecule, so it's not a whole cell, right? A whole cell wouldn't require phagocytosis. So this is some kind of transport that's going to let it in. What is the process of letting this in? It's going to try to get in. It can't get in. What molecule? At least in humans, what molecule is required to stimulate that receptor, that channel, to start showing up in the membrane outside the cell to let the sugar in? What protein? You know the protein, I wouldn't be there's a little couple conversations going on, and that's why you're distracted. It's these distractors, your whole lives are full of distractors. There are people literally trying to get their chapter 8 work together now. I'm about to give you a 0. Because what I'd like you to do is be taking notes on what we're talking about and getting ready for the next test.
I have everything on my board.
That you talking back is not really going to be successful here. So what you need to do is stop with the chapter 8 stuff and focus on the discussion. I don't care if you have what's on the board, because I'm discussing something and explaining to you what you need to know to do well on the test. That's not on the board. That's coming out of my mouth. And if you're busy working with chapter 8, you can't. What are you going to do? So you got this thing's trying to get in. Some other molecule's going to allow for a channel protein, a specific protein it's a carrier protein, is going to come in. But this carrier protein has to have a signal. Something has to make this carrier protein be made by the DNA that's in the nucleus, right? Somehow this channel protein has to be made and be put into the endoplasmic reticulum. We went through the whole process, didn't we? You have the endoplasmic reticulum, you got the rough and the smooth and the Golgi body, and then the Golgi body is going to allow for this little green molecule to start showing up all in the membrane, right? But what is the molecule that tells this DNA, make more of these green carrier proteins that let more sugar in? What is the hormone, the signal that tells the cells of your body to make these proteins to get sugar into the cell? Insulin. Thank you. I know you know it. You learned it. Insulin. Insulin signals the cell to make these channels, right? So that's what lets it in. So the sugar comes in. Oops. Hold on a second. So the sugar's... Oh, you're kidding me? It's a joke. Let's just get rid of it. All right, so the sugar is going to come in to the cell through this little, through this space, right? Now it's in, right? It couldn't get in without what? Without the transport protein. What put the transport protein in the membrane? Insulin. Insulin was the signal. Hey man, we need more transport proteins. Okay? That's what insulin does to every cell, to most cells of your body. So in comes this. Now we got to trap it. Why doesn't it? It'll go right back out. It'll go in and out. What's going to stop it from going out? You have the first enzyme of glycolysis. It's going to add a phosphate, inorganic phosphate. By the way, there's two ways to write phosphate. You can draw it like this, right? Or you can do this. And you saw the PI on the test. Yeah. It's inorganic phosphate. So they attach inorganic phosphate. Now the glucose can get in and out, but glucose with a phosphate on it can't. It's trapped. So that's actually the first ATP is used to do that. It traps it. So the first ATP is used to trap in glycolysis. So now let's take a look at the steps of glycolysis. Do I need you to know that there's 6 steps? No. Do I really want you to in AP biology, you're going to have to learn that. You have to know the enzymes involved. You have to know the enzymes involved. You have to know what the products are. You have to know, for instance, glucose 6-phosphate, fructose 6-phosphate, PFK. You have to know where the ATPs are used. You have to know how many ATPs are used at each step. You have to know how many ADPs are used, how many NADHs are made, how many NADHs. There's no NADPH used in glycolysis. I promise you it's going to be on Friday's test. I'm going to try to trick you. It's not really trick you. I'm testing to see if you're paying attention, right? This NADH is used in glycolysis. NADPH is used in photosynthesis. It's a slight, small difference. But how many NADHs are produced, how many ATPs are produced, all that you're going to have to know about glycolysis. But
You said NAD.
moving on. You can watch the video tonight. Yeah, you should know that. You should go back and read the book. You should have done the homework, right? So, I'm sorry. I have to hold you accountable a little bit. I'm going to go ahead and do that. So let's talk about glycolysis then. What does it look like and what is it? Well, glycolysis, glycolysis, it tells you everything you need to know. What is glycolysis doing? What is glyco? Glycolysis. Come on, guys, use your minds. What is glyco? Sugar. Good. Let's see. You knew that, didn't you? Why did I have to help you with that? So glyco is sugar. You know that. Sugar. Glucose, right? Glyco is sugar, right? Glyco, and then what is lysis? What is lysis doing?
Breaking it down.
Breaking it down, that's right, breaking. What is making? If breaking is lysis, what is making? Synthesis, thank you. So photosynthesis is making sugar, glycolysis is breaking sugar.
And so why, so in with that extra?
No, let's not talk about it, please. It's really complicated, it's not possible. Just all I ask you, when I ask you to do, please do that. And what I asked you to do, I already asked you. So what we're talking about here is respiration. Respiration, that's not, it's cellular respiration. And the first step of cellular respiration is breaking sugar up. Breaking it up. So you have C6H12O6, you have glucose. I don't know if they're going to ask you this, but I'm going to go ahead and tell you that there's going to be an energy investment phase. There's an investment going on, which means you have to put 2 in. You're going to invest 2 ATPs. So you're going to use 2 ATPs in the first half of this. And the reason you're doing that is to break it up. Is you set it up, you're setting this molecule up so that you can break it up evenly. It's almost like when you get a fish. You ever go fishing? Anybody go fishing?
Yeah.
You've seen people go fishing?
Yeah.
Have you eaten fish?
I'm allergic to
Yes.
Do you have eyes on your fish?
No.
Some people do, but... All right, do you have gills? Do you eat the gills? Do you ever see like the scales or the fin? Do you see the fin on there?
No.
No, when you're eating fish, it's all been what? Prepped, right? Before you cook the fish, usually, some people cook it with everything. They've taken the bones out, they've taken the scales out, right? So this is what's going on in the investment. You invest some energy before you get to eat, right? You invest some energy in cooking dishes and cleaning the fish and preparing the pan, right?
cooking dishes.
Jesus, God, help me, Jesus Christ, please.
I'm sorry.
No, you're going to be sorry. You're not sorry yet, but you will be sorry when on Friday. It will happen. It will. I'm just sorry about it. I'm sorry about it now. You'll be sorry about it later. So I'm going to tell you, you invest in order to get something out. So here in glycolysis, you take glucose, you invest 2 ATPs, and your whole objective is to set it up so that you can eat. And so what you get out of the end of glycolysis, what you end up getting is you're putting 2 ATPs in, and you end up getting 2 out. So that's, you lose 2, I'm going to put a -2 if you don't mind. But you gain 4 ATPs for every sugar. You gain 4 for every sugar. So you have a net of what? There's that word net that so many of you keep asking me about, and I keep using it, and I keep defining it. A net of 2 plus 2 ATPs. So that's just out of glycolysis. That's not a lot of ATP. There's still a lot of energy left in that sugar that we don't want to go to waste. And also you produce, the other thing that you produce is 2 NADHs. Now, NAD, do you see a P in there at all?
No.
No. NADH, that's what happens in cellular respiration. Now, the other thing that you produce is at the end, after you've taken this 6 carbon, this is 6 carbon, 6C, right? You take it and you turn it into 3 Cs. And those 3 Cs are pyruvate. Pyruvate. There's 2 of them. Okay? There's 2 pyruvates. Now there's a lot of energy in these pyruvates. That's all your cell can do. Your cell by itself, that's all it can do. It can take the pyruvates out and make 2 ATPs for every 1 sugar. You'd have to eat a lot of sugar in order to make enough ATP in this process to be successful at living the way we live as multicellular organisms. Luckily for us, we have a partner, don't we? What's that partner called?
What?
Wow. Nobody, it's like, people are distracted, even without distractors. You have pyruvate, your cells can't break these down by themselves. They need a helper. What's the helper? What's the partner that we talked about for this morning? All morning. Talked about a partner. Insulin's a protein. We're not talking about proteins. A partner.
He just said insulin is a protein.
I wonder, all right, well, whatever. All right, so the partner is the mitochondria. That ancient prokaryotic organism that was sucked up by our ancestor and became a partner. It became an endosymbiotic partner that now is an organelle. Right now, it's a full-fledged organelle. This thing can take this pyruvate and this NADH, and it can make a lot more ATP, a lot. But it needs electrons, high energy electrons from NADH, right? Just like NADPH, NADH is an electron carrier. Are we good with that?
Yeah.
Okay.
That's like when you, how you explain it, it's like when you need a jump for your car.
Yeah, I could see where you're going with that. I would say it's more like you're the farmer and you're going out and you're getting your potatoes, but you need cash, so you partner up with a restaurant and you provide the potatoes to the restaurant and then they give you cash. Right?
That's how like Bob Evans.
Like Bob Evans or anybody, right? So you're the farmer. So the cell is getting the pyruvate, but the mitochondria needs the pyruvate in order to make the cash. What's the cash? ATP.
mother.
Watch it. All right. So the very first thing that happens is it goes into this mitochondria, this pyruvate and NADH. And the very first thing that happens, let me see if I can. I'm trying to simplify this. Does that make sense? Because if you've done the homework, you know there's a lot. Right?
Yes, sir.
And I could make it go. I mean, in a lot of biology classes, they'd have you like memorize the steps and all that. I'm going to tell you, I'm trying to make it simple, but I guess it's not helping if you're not working. It doesn't matter how simple I make it. But so hopefully you listen. You're taking this inside. As soon as we get inside, I'm going to go ahead and make this small, right? And I'm going to say, let's look inside. So now we're looking inside the mitochondria. Let me get rid of this color and just say, here's the inside the mitochondria. Right in here, the very first thing that happens is pyruvate. Where do those carbons come from in pyruvate? What is pyruvate? Who's been paying attention? Glucose. It's the breakdown product of glucose. There's two of them. There's three. I'm just going to draw it real simple. I'm not going to draw the molecular structure. I'm going to draw it real simple. 3 carbons. There's 2 of them. That makes you how many carbons? 6. So that was in sugar, right? Where did the carbons in sugar come from? Carbon dioxide. That's right. Carbon dioxide. And there were like 8 questions or 9 questions out of 20 on that test that asked that question over in different ways. So if you didn't know that, you better get that in your head. The carbons in sugar come from carbon dioxide. The oxygen released in plants is from water, not from the CO2. The oxygens in CO2 get input in the water as well. I mean the sugar as well, not water. So now, pyruvate, 2 of them. The very first thing that happens, guess what do you think you're going to do with these carbons in pyruvate? Who can guess? Break it into what? Close. All those carbons are going to turn into
CO2.
CO2. The carbons came from CO2 and they return to CO2. So it's a cycle within a cycle within a cycle within a cycle. So pyruvate goes in, CO2 comes out, and then you get the energy put into various places. You can put in acetyl-CoA, then it goes into aconitase and isocitrate and alpha-ketoglutarate and succinyl-CoA and succinate and fumarate and malate, and then back to oxaloacetate and then back to citrate. And it just keeps going back around and around, and just taking out as much of the energy as you can from these. But do I want you to know all that? No, I don't, because it would just give you a headache, and it would give me, I would be very frustrated in the end, and you wouldn't do a good grade, and you wouldn't learn much, right? So I don't really want you to memorize that. What I want you to do is to remember that you're getting the CO2. Where is the CO2 gas produced? The very first step of what we call what? What is this cycle that I just described? I described the cycle. What's the cycle called? It's not the Calvin cycle. No, see, this is where you got to work on this. This is why you get questions wrong on a test. The Calvin cycle made the sugar. Good. Good. That is a cycle, and it is something. It is a thing. It's in photosynthesis, and it makes the sugar. What's the cycle that breaks it down? Krebs that's close enough. It's Krebs. Krebs. But you're right. I mean, you'd get it right on the test, right? Unless somebody thought, well, let me spell C-E-R-B and C-R-E-B and see which one they pick. That would be wrong. But a Krebs cycle is what it's called. The other thing it's called is Tricarboxylic acid cycle, or TCA. And the other thing it's called is citric acid. So all three of these names could be used. I don't know what name they're going to use on the end of course exam, but I can tell you this. It's called Krebs because that's the guy that discovered it. Not crabs, Krebs. C-R-E-B-S. It's called the tricarboxylic acid cycle because guess what's in the cycle? Tricarboxylic acids. Don't worry about it. It's just TCA. Or you might see citric acid because guess what's in the cycle? Citric acid. So it's all the same thing. It's just three different names. Just like there was PI and there was a P with a circle around it.
Yeah.
Don't let the little things, don't get caught up on the little things. All right. So the three are interchangeable. I would know Krebs because it's highly likely they're going to call it the Krebs cycle. But they could call it any one of these. It's fair. It's wrong. They should pick one and stick with it. I agree with you. But they don't. So live with it.
Wait.
So the gas that you exhale, by the way, every time you put CO2 out, where did it come from? From the sugar that your mitochondria, that turn into pyruvate, that your mitochondria in the TCA cycle, Krebs cycle, what have you, broke up the CO2 off. Does that make sense?
Yeah.
Yes.
So CO2 is formed right at the beginning. This is where CO2 is formed. So does the oxygen in CO2 come from the oxygen that you breathe in? No, it does not. No, it does not. It came from the sugar. No, the CO2 here, the oxygen that the plant releases came from water. The oxygen that the plant put into the sugar came from the air. That carbon and oxygen that's in the pyruvate gets put back into the air by the plant in its mitochondria, by animals like us. After we eat, right? We take that sugar, we break it down. So that's the CO2. That's why you breathe out, because that's the leftover sugar. So you're literally breathing out the candy bar you ate this morning. Or the cup of sugar, the sugar that you put in your tea, right? That's what you're breathing out. So that's interesting. So from the Krebs cycle, you're going to produce some things. You're going to produce, from the Krebs cycle, how many CO2s? Let me ask you that. How many CO2s for every sugar? Six CO2s. 6 C's. There's only 1 C in each CO2, so you have to 6 CO for every sugar. 3 CO2s for every pyruvate, right? But there's 2 pyruvates for every sugar. Does that make sense?
That's what I thought you said four.
That's
Four
ATP.
oh.
Yeah, that was ATP. That ATP is being used by the cell. That didn't come in here. You know, the mitochondria doesn't need ATP. It's going to make ATP. It's no problem for it. If the mitochondria is going to make ATP, what is it going to need? ADP and some phosphate. But it doesn't need ATP. It's going to make it. So now it's going to take and it's going to make 6. This is what's coming out of the Krebs cycle. NADHs.
So that's coming out of that?
Out of this, right? It's coming out of the Krebs cycle. And it's going to make something else that we haven't talked about yet, bless you. FADH2, which is what? What are 2 of those? What do you think these 2 are? What do they do? You don't have to know any details. They're carriers, right? They're electron carriers, bless you. And also from glycolysis came what? How many NADHs? 2 NADHs from glycolysis. So you got 8 NADHs going to the next step. And 2 FADHs, and you get some ATP. You make 2 ATPs. So far, there's not a lot of ATPs being made. You have this sugar molecule. You only got how many? 2 ATPs from glycolysis, right? 4. You made 4, but you used 2, so you're net of 2. 2 ATPs from glycolysis, 2 ATPs from the Krebs cycle. So you're only making 4 ATPs so far. But you made a lot of high energy electrons that you could use what? What are you going to use these carriers for? What did we use them for in photosynthesis? Electron transport chain. Because now we can make a hydrogen ion concentration with chemoosmosis, right? Chemoosmosis, I told you in the video that I sent you earlier in class, everything I told you over and over again, chemoosmosis is not the movement of water. They gave you a distractor, they tricked you on purpose. It's creating the concentration gradient of hydrogen ions. They tricked you on purpose. The next step, when I saw, well, I guess can you be tricked by accident? I guess. So oh, it's 10 o'clock, Jesus. All right, get out of here. We'll finish this, we'll finish it. I'll have to collect it tomorrow.